There are three main biomolecules that keep every living thing alive: carbohydrates, proteins, and lipids. And each one is a specialist with a totally different job. First, carbohydrates. Carbohydrates are found everywhere, from bread, to rice, to fruits, to pasta. And at the heart of almost every carbohydrate is glucose. Glucose is a 6-carbon sugar. We number the carbons from 1 to 6, starting at the carbon in the aldehyde group at the top, then moving clockwise around the ring. Carbon 1, C1, the anomeric carbon, the one that decides whether glucose is alpha or beta. Carbons 2, 3, 4, 5, part of the ring structure, each with an O group that can point up or down. Carbon 6, C6, sticks outside the ring as a CH2OH group. Now, what's the difference between alpha and beta glucose? It all comes down to the position of the O group on carbon 1. Alpha glucose, the O on carbon 1 points down. Beta glucose, the O on carbon 1 points up. Now let's look at the major polysaccharides made from glucose. 1. Starch. Starch is the main energy storage molecule in plants, and it actually comes in two forms. Amylose, about 20%. Amylose is unbranched. Because it's just a long chain of alpha glucose, it naturally twists into a helical structure, like a spring. This helical shape makes it compact, but a bit slower to digest. Amylopectin, about 80%. Amylopectin is also made of alpha glucose, but it is branched. These branches make it easier and faster for enzymes to break it down. That's why amylopectin gives quicker energy release compared to amylose. Together, amylose and amylopectin form starch, the carbohydrate you get from rice, potatoes, bread, and grains. 2. Glycogen. If starch is plant storage, glycogen is the animal version. But glycogen is even more highly branched than amylopectin. This extreme branching means enzymes can break off glucose rapidly, perfect for animals that need fast bursts of energy. We store glycogen in our liver and muscles, ready to break down when blood glucose drops. 3. Cellulose. Cellulose is totally different, even though it's still made of glucose. Cellulose is built from beta-glucose, not alpha. Because of this, the chains run straight, not coiled, and they hydrogen bond together into strong fibers. This forms the cell walls of plants. Humans can't digest cellulose because we lack the enzyme to break beta-1, 4-glycosidic bonds, but it's still important as dietary fiber. Now, you may be wondering how the glucose molecules stick together in polysaccharides. When glucose molecules join together, they form a bond between carbon 1 of one glucose and carbon 4 of the next glucose in a condensation reaction. That's why it's called a 1,4-glycosidic bond. In alpha glucose, the O on carbon 1 points down. So when glucose molecules link via alpha 1,4 bonds, the chain gently curves, forming the helical structure seen in amylose. In amylopectin and glycogen, you still have alpha 1,4 bonds in the main chain, but also alpha 1,6 bonds where branches form. So for starch and glycogen, remember, alpha 1,4 makes the chain, alpha 1,6 makes the branches. Cellulose is different because it's made of beta-glucose. In beta-glucose, the O on carbon 1 points up. To form a stable chain with beta-1,4-glycosidic bonds, every alternate glucose must rotate 180 degrees, flip upside down. Why? Because without the flip, the O groups would be misaligned and the bond couldn't form. This alternating flip gives cellulose its signature structure, a straight, uncoiled chain, with hydrogen bonds forming between parallel chains, resulting in strong, rigid fibers.